Okay, then. Final live lesson on YouTube for a little bit. I'm going to be doing some stuff in the evenings. The live lessons will continue. They're going to be slightly different next week, but they're going to be taking place inside the Study at Home Exambulance. Link in the description. It's totally free. Sign into the course. We're going to be just giving our live lessons inside the Zoom room rather than inside a YouTube live because it's just not that convenient for teaching. It's just too much delay. We'll be doing more checking in with you to guide you along the process, to sort of chaperone you through you studying at home rather than us sort of delivering content to you. Also, for year 13s, we're making an announcement today at 4 o'clock on our YouTube live about how your grades will be awarded. We have some more information on that. And yeah, that is something that I'm just looking at the wrong page over here quite clearly. I'd like to yeah, so we're going to be doing a YouTube live 4 p.m. today. Now that is Friday and I don't even know today's date. That's how wild the days are right now. How do I view the front end of my YouTube here? I can see the creator studio, but the comments are even more delayed in the back end. I'm just gonna, I guess we're going to go to my own YouTube channel, can't I? And then I'll see myself as being live. Go to my channel. Okay. So today, yeah, we were going to email all our students this afternoon around before 4 o'clock so that you get the update about the live that we're doing this afternoon to make the announcement about how your grades are going to be awarded a little bit more detail. And we are also going to be doing our Zooms inside and letting our subscribers know about what's happening to the live classes inside of the courses. So we're going to finish off a bit of gene tech today, which I imagine most of you probably haven't quite finished yet. Obviously, if you are feeling like you're not covering all the content and that you feel like you are going to do the exam in the autumn, which we're assuming is going to be September. Obviously, nobody has definitive dates on this yet because we don't even know, it might even be that a second wave of infection springs up and school will be delayed again. But what we do know is that there will be a chance for you to do the exams if you're not happy with your calculated grade. Again, come back to us at 4 o'clock. I'm not going to delve into the details about the grading system. That's what we're going to do at 4 o'clock today. But if you do want to make sure that you've covered all the content, maybe you're going to be doing biology or chemistry or physics or dentistry or medicine or veterinary at university, then you will be expected to have completed the course. And if that's something you want to do, then obviously our full courses are there to help support you so that you can study that all from home. Okay, thanks to all of you who contributed to our Instagram poll yesterday. We'll be picking one of those topics to cover in our Monday. So Mondays, we're going to do them at 5 o'clock. I think we might move them to 5 o'clock. We're going to be doing our regular student check-in to basically answer any sort of ongoing questions that you have about A-levels. So that's obviously right now it's going to be the impact on university applications, UCAS forms, and all your kind of stuff like that. Yeah, we could do some YouTube polls. We're like basically trying to get all the free content that we've put together for you guys sorted and organized first so that we can then sort of leave that running and that you guys get you set up with a bit of a schedule so that you can look after yourselves a little bit. And then we're going to do some more sort of outreach and community stuff. So the stuff that we'll be doing on YouTube in the future, in the next couple of weeks, is going to be more in the evenings. We're going to be interviewing some of our brand ambassadors about their university applications. We're going to be going through talking to some medic students and some dentistry students about their advice to year 12s about how they can help improve their applications, because the applications process is going to be a bit tricky for this year because work experience in hospitals and stuff isn't happening right now. So there's lots of things like that. If you've got ideas that you'd like to see us do on our YouTube channel, people you'd like to see us interview, or questions you'd like to see us answer, then let us know, get in touch, and we can do that for sure. Yeah, the Zoom links will be inside the courses, and they'll be very obvious. There'll be the live class or the live session page, and the links will all be there and put together for you. Okay. Gene tech. What are the topics that are you feel most uncomfortable with across the whole A level? Maybe I can do an impromptu session on what are the topics that you would be most scared about answering questions on? That's kind of what I want to know. Gene tech is something that's typically not very well answered, mostly because most teachers teach it as the last module. They don't really have that much time for it. It tends to get a bit rushed. They don't tend to do many exam questions on it, and then it's just generally not very well received. Cellular control, gene tech, variation, evolution, inheritance. The good news, inheritance is tricky. The good news is, if you understand it, then the questions get a lot more straightforward, but you've got to do quite a lot of legwork or brain work to get your head around the inheritance and really understanding meiosis. So gene tech, gas exchange, lots of you saying gene technology, kidneys, liver, so homeostasis, muscles, more kidney, more inheritance. So I'm seeing sort of like, broadly speaking, it's gene technology, inheritance, and homeostasis. Okay, transcription translation, again, they're quite procedural. The more complicated with transcription translation will come in the control of gene expression, what happens normally in terms of production of a protein, and then how you can alter those sequences to affect the outcome of the protein. Okay, so so Jacqueline, I'm fine with the content, but it's the questions, analyze data and use your own knowledge. So are you an Edexcel student? Analyze is a question that Edexcel use more than anybody else. The exam guide of our courses, the exam guide would be that's why we have two courses. The content guide teaches you the content. If you're missing understanding, you're missing a complete set of notes, content guide is going to deliver that for you. If you're like Jacqueline, it's like, actually, I kind of understand all the bits and pieces. I don't need to create more notes. I need to understand how to apply my knowledge to the exam questions. That's exactly what the exam guide is there for. Today, we're going to do some gene tech, I think. That's what was planned, and it's one of the topics that has been standing out there as one of the ones that you'd say that you'll have the most difficulties with. And maybe we'll do some questions today. Maybe we can just do some pot shot questions. My iPad is just charging. It was pretty much out of juice. I'll pull up a question on my computer and share it with you. And then I'll maybe, if I need to delve into it, I'm sure my iPad will have 10% by now, and it will be enough to see us through the rest of the session today. So let me find a question. I'm going to do a gene tech question. Any specific aspects of gene technology? Yesterday we looked at a bit of PCR, so we could start off with a PCR question, or we could look at an electrophoresis question. Give me a gene tech topic to search for. It's fine if I put these on the same window. Okay, more PCR than electrophoresis. So let's find some PCR. Oh. Okay, it's a really simple one here. Again, they're going to be down the bottom of the screen because I have the mark scheme answers on this document as well, and I want to test you before showing you the mark scheme answers. Where's my PDF viewer? Okay, so the question, sorry for it being right down the bottom, question is that a small amount of DNA can be extracted from mitochondria. We looked at cell organelles earlier in the week, so mitochondria and chloroplasts have DNA, basically it's the same as prokaryotic DNA. It's singular loop. It is not in a nucleus. It does not contain introns. It's shorter than regular eukaryotic DNA. So a small amount of DNA can be extracted from mitochondria, and DNA sequencing is used to try and find a mutation. So you're looking for a mutation inside the DNA from a mitochondrion. From this sample, how would enough DNA be obtained for sequencing? How would sequencing allow the identification of a mutation? So we've got 2 marks and 2 bullet points. So what do we have to do for those things? So it's a relatively simple, straightforward one now. I'm just going to pause my screen share. I'm going to find another question because, oh, no, I can't just yet because you'll need to see the marks. Can you? Okay, well, I can just tell you the answer to that one. You can just take my word for it. Okay, this is probably it's a nice little summary one. PCR to amplify the DNA. What about finding the mutation then? Identification of the mutation. You're going to compare the base sequence of the DNA that contains the mutation to, yeah, you could say, I think normal mitochondrial DNA or mitochondrial DNA known not to include the mutation. You're going to compare it base by base. So that's exactly what you would say. Okay, so let's, I'm going to jump to this question now. So one way to detect and measure accurately the amount of RNA, here we're talking about RNA in a tissue sample, is by using RT-PCR, reverse transcriptase polymerase chain reaction. So we are assuming we know what reverse transcriptase does. What does reverse transcriptase do? Convert messenger RNA. It'd be nice to include the C, the complementary DNA. If we asked that, if you were asked as a 1-mark question, what does the reaction does reverse transcriptase catalyze? Then you'd expect it to be give the little C for complementary DNA. Nice. Okay, so this process uses a reaction mixture containing the sample for testing, which presumably is going to contain the sample RNA, reverse transcriptase, the enzyme, DNA nucleotides, primers, we've talked about all this week, DNA polymerase, the enzyme, could be Taq polymerase if you're going to run it at high temperatures, and a fluorescent dye. The principle behind this method is shown in figure 8. The amount of DNA amplified by DNA depends upon the amount of RNA in the sample. The dye only fluoresces when bound to DNA, so it does not dye when it's bound to RNA. Well, it won't bind probably to RNA, but it will only fluoresce when it bounds to DNA. The intensity of the fluorescent light emitted increases as the PCR produces products accumulate. So the more, again, here it says PCR products, but what is the product of the PCR? It's going to be DNA. So it's just saying the more DNA there is, the higher in the intensity of the fluorescence. Explain the role of reverse transcriptase in the process. So this is the question. And I would think, to be safe, you would say to convert to the messenger or the RNA. We don't, it's just been stated as RNA here. So it doesn't have to probably include the messenger RNA. So I think you would be allowed to say RNA to complementary DNA. Okay, so they've been nice here. The C and the M are both in brackets, so they're not mandatory. So it makes DNA using RNA. Except this, reject transfer RNA, but that's fair, I think. Okay. Explain the role of DNA polymerase. The enzymes, I'm sorry, right down the bottom, the answer is right there, so I can't show it to you. What's the role of DNA? Again, we've got 1 mark. We don't have to go into crazy detail here. What does DNA polymerase do? You're expected to know all these enzymes: DNA polymerase, reverse transcriptase, DNA ligase, RNA polymerase. What other specific enzymes might possibly come up in this section? It's basically the ones. Okay, forms the phosphodiester bond. A lot of you naming that nicely. It attaches the nucleotides, adjacent nucleotides together to form the phosphodiester bond in a condensation reaction you could add. Joins nucleotides to produce complementary strands on DNA. I like you guys including phosphodiester bond in there. I think that's tip top. If you are writing a longer answer on that, that phosphodiester bond will probably be a separate marking point, so an additional mark. So I've trained you well. Actually, a pretty straightforward answer, that one. Okay, as we scroll down a little bit, again, the mark scheme's just gonna be down there. Any DNA in the sample is hydrolyzed by enzymes before the sample is added to the reaction mixture. So remember, what were we starting with in the compound here? We were starting with RNA, and we were going to then make the RNA into DNA, and then we're going to do our PCR from that. So why would you want to hydrolyze any DNA in the sample to begin with? 2 marks. Yeah, to prevent contamination, you don't want to copy this other DNA. You want to copy the RNA. So you want to destroy, in the beginning, you put RNA in and some contaminated DNA that you don't want. So you only want to copy the RNA, which means you want to destroy all the DNA at the beginning so that when you actually start producing the copies, you're only copying the RNA. To remove any DNA present, as this would also be amplified or replicated. So Bape, you said so only the DNA sample is amplified. It's completely the right point, but it's RNA that is being, it depends on how you phrase it in the actual exam, but it started with RNA. So only the RNA sample is amplified would definitely be fine. To prevent the contaminated DNA being amplified would also be fine. Okay, so any DNA in the sample, okay, so that kind of comes back to the word sample. The sample for testing is here. What else is in there? The enzyme, DNA nucleotides, but not DNA polymer, single nucleotides, primers, DNA polymerase, and the dye. So it's saying why in this, the sample contains RNA that we want to copy. There will also, it's saying that there's also some DNA in here as well. But we don't want to copy that DNA, we want to copy the RNA. So why would you want to hydrolyze any DNA in that sample? Well, you'd want to make sure that any DNA is destroyed so that we only do the PCR on the thing that we want, which is the RNA in this circumstance. So with these questions, one of the core themes is keeping track of you can't remember all the words in the question, of course not. But what you can do when you do that question analysis that I go through in the exam guide is like, what is important here? Which bits do I need to save so that I'm aware of this when I'm writing my answer? Otherwise, it's like, well, you don't know what's important. Yeah, we've gone through that. Must be the idea of removal or destruction, so that it doesn't use the DNA as a template, that it does use the RNA as a template. Okay, so, and then a bit of graph analyses. I don't know why my scroll speed has gone so slow on this mouse. Figure 9 shows the results from the RT-PCR to detect RNA in 2 different samples. So we had sample A and sample B. So these are 2 different independent PCRs. And we've then got the fluorescence. Remember, the fluorescence is being produced by the dye attaching to the DNA after the RNA has been copied in this process called RT, reverse transcriptase PCR. So quantitative comparison can be made of the amount of RNA in samples A and B. So quantitative. This involves determining the number of cycles required to reach 50% maximum concentration of DNA. So this 50% maximum of DNA is called C. The amount of RNA in a sample can be measured as 1 divided by C. Use this information to calculate the ratio of RNA content in sample A to the RNA content in sample B. I'm going to definitely need my iPad for this, so let's hope it's got enough juice in it. Let's go. I think I'm going to need, I'm going to just take a screenshot of this. I can just. I'm going to send it to myself so I can actually just draw on the question on my iPad. Okay. So I'm going to let you start working on that. So what is the first thing that we need to do, or the first things that we need to do? Whilst I just shoot this image over to myself. Okay. So I've just copied the question over here. Actually, what's going to happen there? Whoop. Oh, awesome. There, so I can write on it. Wonderful. Okay, so we've got 50% of maximum. So what is maximum, I think, is our first question. Maximum is up here. So what value does this correspond to? 0.46, 0.47, 0.48. So 0.48. Okay, well, 1 o'clock. Okay, we'll do 1 o'clock. We still need to email everyone, Ronnie. So that email needs to go. It can go at 12:30, but we need to, as soon as I finish here, I'll come and chat to you about that. Let's finish this question. 0.48 divided by 2 gives us 0.24. So then we can then take this line across again from 0.24. And then we're obviously going to read that down. Where does that cross the line? Okay, this one is, it's quite difficult to do on. No. Let me draw a line. Maybe I'll just do it from the. Okay, so we're basically trying to find the number of cycles. So we're getting close. So wrong pen. How many cycles is this? 15, 16, 17. I think probably sample A is going to be 18 cycles is 50%. Cycle B is 25, 26, I think 27 cycles gives us our 50%. So now what do we need to do? The amount of RNA sample can be divided as 1 divided by C. So we could do, so 1 divided by 18 in 1 divided by 27. So what do we get if we punch those numbers in? Well, let's maybe just do the numbers underneath. 1 divided by 18, it'll be more obvious. 1 divided by 18, put it in decimal form, 0.055. Recurring, 1 over 27. 0.037. So these as a ratio, we're going to divide by the smallest one to get the ratio. So that this one divided by itself is going to equal 1. Anything divided by itself is 1. And you're going to just choose the smallest number at that point. So then we're going to do this number divided by this number, and that's going to give us no, it's not right. 1 divided by 18 divided by 1.5. And we want to give this is sample A, and this is sample B. So let's just check. Use this to calculate the ratio. 1.5 to 1. Now the question you've got to ask here is that do we want to give this as 1.5 to 1, or do we want to give it to 1 to 0.66? I've been ignoring the chat. Does it have to be a whole number ratio? Well, one of them is going to have to be represented to 1. Yes, one of them will always be 1. So the only thing I'm not 100% sure on right now is like, do we give this as, we could say it's 1.5 to 1 as our final answer, or we could convert this number to be 1 and then make this number less than 1, which would be 1 divided by 1.5, which I think is 0.66. But I don't know. I don't feel like we need to do that meddling here. I feel that's unnecessary. I feel like 1.5 to 1 is enough. If Ronnie's still there, I don't know if there's a mathematically correct way of doing that. One of them is going to have to be a decimal either way. No, you would always, as a whole number, no, you would always do it something to 1. One of these numbers must be 1, and the other number is either going to be a bit bigger than 1 or a bit less than 1, depending on which way around we do it. Okay, so I can go back to the, does anyone follow the method? Anyone have any issues with what we did there? So step 1 was find out what 100% was, 0.48. So what's 50% of that is 0.24. We then read that across and then find out how many cycles for each of those that was. C is 1 divided by that. So it was 18 cycles for A, so 1 divided by 18. It was 27 cycles for B, 1 divided by, so this is our ratio, but we then just have to plug the numbers in. And if we put them as decimals, we get this. And to get anything, to make this into 1, we divide by itself. I nearly sneezed. Full bore at you then. I should go and wash my hands right now, but I'm pretty safe. I've been in isolation for, how long have I been in isolation now? It seems like forever. I did a week in Australia, then I got on some long haul flights, and now I've been here nearly a week again. Okay, I'll go back to the mark scheme so that you can see that I'm not just making it up. Ratio in the answer of 1.4 or 1.5 to 1. Okay. Note, not expressed to 1, correct range may be shown in different ways. So you get 1 mark for saying 3 to 2. How do you know which order to put it in? As in, how do you know the 1.5 comes first? Is that your question, Edith? Because in the question it says the ratio for A colon the ratio to B. So A must come first. Suggest one reason why DNA replication stops in PCR. Why does it ever end? Simply, you either run out of nucleotides or you run out of primers. It means not why is it, okay, so, okay, well, not why does one cycle stop, is why does it all stop? Yeah, there's no, your limit, the enzyme is reused. So you wouldn't say that the enzyme runs out because the enzyme is reused. PCR would continue with or without the fluorescent dye. You just wouldn't see the results so clearly. You wouldn't see the fluorescence changing, but the PCR would continue. This is asking why the replication stops. So you're running out of primers or you're running out of nucleotides. I mean, it does eventually say, eventually the enzyme denatures. Okay, so scientists have used this method, reverse transcriptase PCR, to detect the presence of different RNA viruses, very apt for right now, in patients suffering from respiratory diseases. This is a paper from 2017. I didn't know this was a question, but this is perfectly poignant. The scientists took a variety, produced a variety of primers for this procedure. Okay, so let's analyze this question. I don't know where this is going immediately. Have used the PCR method to detect the presence of different RNA viruses in patients suffering from respiratory diseases. The scientists produced a variety of primers for this procedure. Okay, that's not that difficult a question. Exactly, Edith. You've got different viruses. They have a different RNA sequence. Therefore, you need a different primer for each one. Base sequence differs. Complementary primers are required. Nailed it. I think that's the end of this question. It is. Okay, do you want to do one more? Or do you want to call it a day? You can let me know. Use my iPad out of the way. In fact, I might be able to find a question. If I do another question, I might be able to find it on here. Nearpod, I might try and do some Nearpoddy stuff on YouTube at some point, but effectively it takes hours to set it up, and then it's actually pretty inconvenient to use really nicely. Like only the iOS app allows you to take photos. I really would like you to be able to handwrite your answer, take a photo and share it with me, which would be great, but that's only on the, it doesn't work on Android, for example. Okay, so let's find Let's find an OCR question here on let's try this one out. See if I can risk a bit more brightness on my iPad because it's pretty dark right here. Okay. Not the best question. It's a good start to the question, but then it goes into OCR specific stuff only. Okay, this is a bit more general question. What number is this? Question 7. Okay, let's try this. Okay, let's do the start over here. I tell you not to start at the just any old part of the questions. I'm just going to go through it. We've got like a family tree right here, looking at question 7. Evolutionary relationships as phylogeny. We've got different mammal species. We're not going to do this, but okay, so evolutionary relationships, we can look at proteins and the sequence of proteins, the albumin, hemoglobin, or cytochrome C. I'm just giving us context for the question. The roles of 3 proteins are as follows. Okay, so basically the bit I want to look at is over here, but I was just making sure that there's nothing that's going to influence my answer in the early part over there. Okay. Research on another protein from the giant panda was carried out in 2008. The protein was called crystallin. I should do this in blue, I suppose. So it's a protein, it's called crystallin. It's found in the lens of the eye. It has a sequence that has been highly conserved. What does that tell us in all mammals? What does it tell us if a sequence has been highly conserved? The steps in the procedure used in the study are summarized here. Reverse transcriptase is used to make the crystalline complementary DNA from the panda's messenger RNA. If a gene is highly conserved amongst the mammals, it means it is really important. It is, without it, you don't stand a good chance of surviving. It is well preserved, because if it's not that important, you get a mutation to it, it's inherited. If it's not that important to it, that mutation might just continue in the population, because it doesn't really matter whether it is mutant version or not. If it's really essential that that protein works and functions, if you have a mutation that causes it to become non-functional, that organism is likely to die. And therefore, only those that have the original faithful copy are passed on. Therefore, it is saved or conserved over generations. So if something is reserved or conserved, a gene or a protein, then it's really useful. It's unlikely to change that much over time, just because without it or changes to it are usually lethal or bad for you. The panda crystalline complementary DNA undergoes a process to increase, okay, look at this, a process to increase the quantity of complementary DNA. What is this process? In my head, I'm just thinking, bing, here's a mark, or going to be a mark. I don't even know what the question is yet, but boom, it's PCR, of course it is. And they've gone out of their way to not say PCR. Complementary DNA is introduced into bacterial cells using a plasmid vector, and the bacteria are grown in petri dishes. The crystalline protein is harvested from the bacteria and separated from other sized proteins on a gel. The crystalline protein and complementary DNA are both sequenced by automated sequencing methods. So sequencing a protein means you're looking at the sequence of the amino acids that form up the primary structure. Sequencing of the DNA is obviously each protein, each amino acid is going to be coded for by a sequence of bases. Identify the technique used to carry out steps 2, 3, and 4. So step 2, you've already told me, we're increasing the quantity of DNA, PCR. Step 3, the technique to introducing complementary DNA into a plasmid. It's a technique, so it's not going to be a restriction enzyme. I don't know what I've just done there. I don't know how far I've just traveled in this paper, but I might be able to do this. I'm not 100% sure on that myself. I'm just trying to get my head around it. Or in which case, you're not going to know every question all the time. Sometimes it will come to us a bit later on. So let's just move on. DNA is introduced into so the process. I think this is an OCR. This is an OCR paper. So it could be electroportation or electrofusion to give the name of April, but that's really only an OCR term because they do cloning, animal cloning, where that key term pops up again. So that's the only thing I can think of as a process for there, but I'll check the mark scheme and we'll double check it. And 4, the protein is separated by size. How would you separate molecules by size? That is electrophoresis. It works for proteins as well as it works for DNA. It will separate things according to if they've got a charge and R groups have charges, then you can make that happen. Okay. The panda crystalline protein obtained was 175 amino acids long, corresponding to 528 base pair of complementary DNA. Explain or give reasons why a protein that is 175 amino acids long is coded for by 528 base pairs of DNA for 3 marks. So how many bases? AQA have asked questions about electrophoresis of proteins. Probably a suggest question, but yes, they have done it in the new spec. I don't know why you're not seeing what I'm seeing. I'm going to stop my share and just reshare it because you're not seeing everything that I've written. And now you've got a black page. Let's try that 3rd time lucky. Okay, so what I would do is I'd do the number of amino acids. Why can't I? Oh, it's because it's across the page. I'm going to do 175 multiplied by 3. And it might not come in exactly that number. 525. Okay. So then we've got plus 3. I was kind of expecting 6, to be honest, but equals our 528. So what's the extra 3 about? So there's 175 amino acids, each one coded for by a triplet. So there's 3 bases per amino acid, which gives us 525 bases, but we've got 528. So there's 3 left over. So I was expecting 6 different because I was expecting a start codon and a stop codon. This, we can take our pick. I'm assuming it's the start codon, but it could be the stop codon. Everyone looks like they're on it. I think they will allow either. I wonder if I can find out. This is to give you a bit more of a nudge in the right direction. Okay. So this process, I've just checked the mark scheme over on that. The process that they for step 2, what's your bets? See if you can. I would have got it wrong. So see if you would have beaten me for the process for step 3. Introducing DNA into a cell of a different organism. It's a broad term. It's the name of this topic, more or less. Genetic engineering is what is in the mark scheme. Or genetic modification. I mean, like, that's something that we all know, right? We all know that, but the question is not clear enough. And that's what I say. You're not going to get every question right. Even with my experience doing thousands of these things, I was looking for a much more specific process than that. And I was trying to name the process by which the plasmid was inserted. And the only thing I could think of was so that electrofusion, electroportation, which is an OCR only term, and it's mostly used in cloning and not really used in gene tech. But if you got it, then well done. And if in doubt, just give the most sensible suggestion that you can, and just take comfort in the fact that an A is about 55%. So you can get 45% wrong and still get an A. So, okay, I will share the mark scheme. In fact, I think I can do that in here whilst I'm except gene cloning or transformation, electrophoresis. Okay, so the triplet is 3 bases is 1 amino acid, which gives 525. 3 bases are stop or chain termination codon. Interesting. So they want stop codon only. Just a little bit of learning we can do there. So yeah, separating proteins or DNA according to size or charge is electrophoresis. Okay, so update, not 4 o'clock anymore. We apparently can tell you as of soon. So we'll come back at 1 o'clock. We will sort of replace the year 13 maths live today with the announcement about how your year 13 grades are going to be announced. Are you going to join us there? Are you not? Are you going to come back for that? The updates. So we'll be announcing at 1 o'clock. Yes, we do have some updates for you. Okay, well, we'll see you at 1. See you then.